Good evening. So, right, so there are a few questions I see. Uh, can you switch on your mic and then we can talk on the questions if that's not a problem?
Sir, good evening, sir.
Yeah, tell me, what is your question?
Sir, can you explain the concept of that angle formula, sir? Psi angle. Week 2 assignment, you are asking one question, sir.
You're asking on some 3 prime to
And
5 prime.
week 1 assignment, we are having 3 prime, 5 prime conversion, mRNA, dRNA.
Yes, mRNA, tRNA, right? So, you want a clarification on that,
Yes, sir.
right?
That conversion type I'm asking, sir. GC, AT. Yes,
Okay,
yes,
so
sir. Yes, yes.
right. So basically, when you means when there is a translation, a transcription from a DNA to a RNA, we use basically the 3 prime to the 5 prime strand, okay, of the DNA. So you know that the double-stranded DNA has two strands, right? A 5 prime to the 3 prime and the 3 prime to the 5 prime, right? Uh, am I audible? Hello, am
Sir,
I
pardon,
audible?
sir. Yes, sir.
Yeah, I was telling that when we think of a DNA, it's double stranded, so it has a 5 prime to a 3 prime and a 3 prime to the 5 prime strand, right?
Okay, sir.
And when we go on to
Yes,
the
sir.
transcription, that is the conversion from a DNA to an mRNA, we use means the
Okay.
RNA polymerase, the DNA polymerase. So basically, for the transcription, the 3 prime to the
Okay.
5 prime is used, based on which the mRNA is produced. So the starting material
Okay.
for the transcription is basically the 3 prime to the 5 prime, from where the sequence from the of the mRNA that you are getting is basically, means when we talk like strand wise, it is a 5 prime to the 3 prime, okay, such that you get the strand as a 5 prime to 3 prime, so the reverse strand is being used in the DNA for the transcription process. And now based on the mRNA that is produced, then in codons of triplets, we actually get the amino acid sequence.
Okay, sir.
Okay, was this your doubt or was it something else?
And sir, this is my doubt, sir.
Okay, so it's clear
Uh,
now.
yeah, yes, sir.
Okay. And the other thing that you are asking me is how much percentage of problem type questions. So
Yes,
I
sir.
don't
Yes.
understand by what is the problem type questions. Means, do you
Yeah.
mean by NAT type, MSQ or MCQ?
Sir, MCQ I'm asking.
So most of the questions are basically MCQ and MSQ only.
Okay,
There is
sir.
no NAT type of questions, or there is nothing questions like you have to write the answers by typing or something. It is just an MCQ or an MSQ, it will be.
Okay, sir. Okay, we have to choose the options only.
Yes.
Okay, sir. Okay, okay.
For the previous years, like this course is quite new, so for the last year, I don't think there were any like typing questions, because you don't get calculators, you don't get things like that. So max to max, you can get MSQs, not MCQs. So
Okay,
there
sir.
will be MSQs, but that's just you have to tick the correct option only.
Okay, sir. Okay, sir. Is it enough to see the assignments only? No, week 1 to 12 assignments and your lecture notes.
Yes. It is based on only the assignments and the lecture series. It's nothing like anything else.
Okay, sir. Okay, okay. Thank you so much, sir. Okay.
So today we will mostly talk about the immune system. That's the host defense mechanism. So immunology basically started in the 15th century, where the diet crusts from smallpox were either inhaled or inserted into small cuts in the skin, and that is what we had named it as variolation. Now, after that, in 1796, Edward Jenner demonstrated that inoculation of the cowpox protects against the small pox, which was a lethal disease at that time, and he termed that as vaccination, which means from the Latin vacca, meaning cow. And after that, Louis Pasteur's hypothesis expanded on Jenner's work by developing the principle of attenuation. He hypothesized that using a weakened or an attenuated version of a pathogen could protect against a full strength disease. And this led to the development of vaccines for rabies and anthrax, moving into the field of modern immunology. So you can see over here that this is how the smallpox officially eradicated in 1980, and this was an image by the World Health Organization. So the trend shows that smallpox was endemic in over 30 countries, and following what the graph shows, so it started in 1967, and the cases literally uh, increased, and then after the development of the vaccine, the cases started to decrease. So the first insights into mechanisms of immunity were Eli Metchnikoff discovered phagocytotic cells that ingest the microbes and the particles. So it indicates that cells confer immunity. And then we had Emil von Behring and Kitatso, who discovered blood sera could transfer immunity, that is, liquid of blood confer immunity. Now, there are two things. What is the difference between serum immunity and the cellular immunity? So, cellular immunity was observed, was introduced after Metchnikoff observed the phagocytic cells like the macrophages, and this immunity is conferred by the actions of the specific cells in the body, whereas the serum immunity was by the other two scientists, who conferred it by substances that was later identified as antibodies found in the liquid part of the blood. These are the sources of cellular components of the immune system. So what are the things that we basically have over here? So if we start with, we have on the top is the bone marrow. So this is the primary site of hepatopoiesis. It generates from the progenitor cells for both the myeloid and the lymphoid lineages. Then we go into the upper branch, that's the thymus. Some progenitor cells migrate from the bone marrow to the thymus to mature into the T cells. We move on to the blood in the middle level that acts as the transport system, carrying mature and immature immune cells to various parts of the body, followed by the lymph nodes and the spleen. So these secondary lymphoid organs are where immune cells often congregate to monitor for pathogens and initiate adaptive immune responses. And finally getting on to the tissues, which belongs to the bottom level. So the final destination for many effector cells. So here the cells like macrophages are derived from the monocytes, and the mast cells performs the specialized functions in the defense and the inflammation. Now, moving on to the immune cells of and the lymphoid organs. So the yellow portion that we see is basically the primary lymphoid organs, and the blue is basically the secondary lymphoid organs. So what are the primary lymphoid organs? It's the bone marrow that is located inside the bones. It contains blood forming stem cells. It is the site where the B cells, innate immune cells and all the other blood cells are created. And thymus, it's a gland located in the upper chest. It is the specific site where the T cells migrate to mature and become functional. The secondary lymphoid organs, you have the lymph nodes, spleen, tonsils and the adenoids. You have the Peyer's patches, appendix, lymphatics. So the T cells matures in the thymus where the B cells are maturing in the bone marrow, right? And if we talk about the Peyer's patches, so these are small masses of lymphatic tissue that is found in the ileum region of the small intestine. You know about the appendix, the tonsils that are located in the throat and the nasal cavity. So that is the first sign of line of defense against the inhaled or the swallowed pathogens. So most of you might be having the problem of tonsils, swallowing tonsils during an early stage where it was the primary sign of fever, then cough, cold and cough. Right. Now, okay. So these are the different immune systems and the functional organs that are part of this immune system. So we know it's a very complex system where skin acts as the primary physical barrier to prevent the pathogens from entering the body. You have the lining of mucus, which is a physical barrier. Secretions like the tears, mucus, which are antimicrobial. The blood cells and the vasculature, which is the white blood cells. The bone marrow, that's the source of hematopoietic stem cell. You have the lymphatic system and the lymphoid organs. So most tissues have resident immune cells. And the immune system is the body's defense mechanism. So a complex system that encompasses every organ and compartment of the body. So the main objective over here is to seek, identify and kill the invaders. So that's the primary objective. It's highly adaptable and can act against pathogens ranging from different sizes and distinguishes between self and foreign and mount to response to only foreign while preserving the self. Okay. So how does the immune system generate almost infinite variety of cells and substances? So what it first does, it recognizes the foreign particles. There is an effector response to eliminate and neutralize the foreign particles. So what happens over here, like when a person becomes infected by a disease, the body builds immunity against that particular disease, which is a natural immunity. And the person can also build a resistance to a disease following the vaccination. That's an artificial immunity. Now, when an infant receives the mother's antibodies, it's natural. When a person receives antibodies, say after a snake bite, the gamma globulin injection, so it's a artificial one. And then the other sign of recognition is based on memory. So upon the exposure, so say you are affected by a particular kind of a disease for the first time, but you have not taken either any kinds of immunity, it's just naturally it has healed up. But the second time when the exposure to that particular infection happens, then there will be certain antibodies that will come up and help you out. So that's memory. That's based on memory. So you have to take certain vaccines only a few times because the other things is simply based on memory. Okay. So the protection against pathogens relies on several lines of defense, like you have the anatomic barriers, you have the say complement antimicrobial proteins, you have the innate immune cells, then you have the adaptive immunity. So mostly what we need to do are what are the functions of these. So anatomic barriers is the first line of defense. They act as physical and chemical walls that prevents the pathogens from entering the body's internal environment, like the skin, oral mucosa, you have the respiratory epithelium and the intestine. Then the antimicrobial proteins, so if the barriers are breached, these proteins immediately attack the pathogens by punching holes in the membranes or attacking them for destruction. The innate immune cells are the first responder cells that recognize general patterns on the pathogens. They swallow germs, phagocytosis, or release chemicals to kill the infected cells. And finally, the adaptive immunity, which is basically the B cells and the T cells. So they are highly specific. It learns the identity of the particular pathogens first, and then it creates a long-term memory, allowing for a faster and stronger response if the same pathogen ever returns. So it's more like a memory immunity. Now, the host defense mechanisms where the innate and adaptive immunity plays an important role. So, in response to the pathogens, the vertebrate immune system uses two connected systems. The innate immunity, which is inborn, so it has a faster response. It impedes the infection immediately. It does not require. It requires no memory, so it responds the same way every time it encounters a pathogen. And for the immunities, Bruce Butler and Jules Hoffman were honored for their activation of the innate immunity work. Whereas on the other hand, if you take adaptive immunity, it's acquired during the lifespan, so it's slower response. It stops the infection and clears the pathogen. It has memory, uh, and the awardees were Ralph Steinman was honored for his discovery of the dendritic cells and its role in the adaptive immunity. Right. Now, this is important because here we will discuss about the innate and the immune response in context to a typical acute infection. So over here what we see is uh, it outlines. So this acute infection is being outlined with the timeline and the intensity of the body's defense mechanisms from the initial entry of a microorganism to the final clearance of the pathogen. Now there are different phases of immune responses. So first is the establishment of the infection where there is a trigger point. So this begins at the entry of the microorganism. So the innate immune system acts immediately. So the level of microorganism begins to rise as the pathogen replicates, and uh, the innate immune response system is the first line of defense. Going on to the second phase, that is the inductive phase. So over here there is a threshold. As the level of microorganisms increase, it eventually passes a threshold level of antigen to activate the adaptive immune responses. This triggers the adaptive immune cell system, which is highly specific to the pathogen but takes more time to immobilize. Now the effector phase is, now the curve starts to take reaches a constant phase, and it has reached its peak. So the combined efforts of the innate and the adaptive systems successfully begins to lower the level of microorganisms. And finally, the memory phase, when after the pathogen is cleared, the active responses subsides. The memory maintains a immunological memory, that is the T cells and the B cells, which allows the body to respond much faster and more effectively if the same pathogen is encountered again in the future. Now, losing the innate arm of the immune system results in more disastrous outcome than the losing the acquired arm. What does it mean? So, say for example, in a normal mice and humans, that's for the green line. So both the innate and the adaptive immune systems are functional. So the outcome over here is that the infection is initially controlled by the innate defenses, the macrophages, neutrophils, followed by the adaptive system, the T and the B cells, which eventually clears the pathogens. This is the yellow line that is happening for the normal mice and the humans. For the green line or the cyan line, whatever you say, so these individuals lacks the acquired immunity, where the T and the B cells are missing, but they still have a functional innate immune system. So the primary stages should be able to avoid, but it does not build up a memory. So the innate system can keep the initial growth of the microorganism in check for a while, but because there is no adaptive cleanup crew, so to provide specific long-term clearance, the infection persists or grows slowly over the time. And this is serious but often manageable in the short term with medical intervention. The last one, where it lacks the innate immunity. So these individuals lacks the first line of defense, neutrophils, the PMNs, and the macrophages. So without the immediate responders, the microorganisms count explodes exponentially with the very beginning. The adaptive immune system takes a lot of time to activate, but by that time they normally kick in, the pathogen load is already lethal, and this can lead to more disastrous outcome, finally death. So in response to the pathogens, vertebrate immune systems use two interconnected systems that we are talking about, the innate and the adaptive immunity. So the response time is hours to minutes for an innate and days for adaptive. For specificity wise, it's limited and fixed, whereas adaptivity, for adaptive, it's highly diverse, adapts to improve during the course of immune response. The response to repeat infection is same each time, but adaptive, it's more rapid and effective with each subsequent exposure. And the major components are the first line of defense, that's the skin, phagocytes, pattern recognition, molecules, PRMs, and for adaptive, you have the T and the B lymphocytes, antigen specific receptors, and the antibodies. So these are the innate immune system. I don't want to go into the depth of it. Now, this is important to understand. What happens after a breach at the epithelial barriers, such as the skin, takes place? So we can see that the intrinsic epithelial barriers to infection, where this is the first line of defense, it includes the skin and the linings of the respiratory, gastrointestinal, and the urogenital tracts. So these surfaces provides mechanical protection, a physical wall preventing the entry, chemical protection like the secretions like mucus acid in the stomach and antimicrobial peptides, and microbial protection like a good bacteria that compete with the pathogens. Now, adherence to the epithelium. So, if the pathogens manage to bypass the surface protections, they attempt to stick or adhere to the epithelial cells. So, at this stage, pathogens use specific surface proteins to bind to the host cells. Resident immune cells like tissue macrophages and the dendritic cells are already stationed just beneath the surface, waiting to detect these invaders. Now, when the pathogens successfully break through the epithelial wall and enter the underlying tissue, inflammation begins. The barrier is physically disrupted, allowing more pathogens to flood in. The local environment changes to try and trap the infection at the site of entry. Once inside this tissue, the innate immune system kicks into high gear. So the macrophages begin to eat the invaders. Blood vessels nearly becomes leaky, allowing more white blood cells and fluid to enter the area, and the dendritic cells picks up pieces of the pathogen to carry them away to the next phase. And finally, if the local innate response cannot contain the threat, the adaptive immune system is activated. So the dendritic cells travels to the nearest lymph node to present the pathogen signature to the T cells and the B cells. Now this trigger a targeted attack, producing specific antibodies and specialized killer cells to hunt down that exact pathogen throughout the body. And this stage is also creates immunological memory, so that the body can respond much faster if the same breach happens again in the future. Right now, so what is an inflammatory response? So inflammatory response is a fundamental type of a response by the body to injury or localized infection that is characterized by the classical signs of vasodilation. So the blood vessels widen up, this increases the blood volume and flow to the injured area. Now there can be increased vascular permeability, that is the walls of the blood vessels becomes leaky, fluid or plasma escapes from the body vessels into the surrounding tissue resulting in swelling. And then there can be inflammation due to heat, redness, swelling, pain. So leukocytes enter the tissue from the local blood vessels and halts or clears the potential threat. Now transition from innate to adaptive immunity. So what happens over here is the transition from the immediate non-specific immune system to the adaptive immune system is bridged by the antigen presenting cells. So these cells ingest the pathogens and show pieces of them to the T cells to trigger a targeted response. So you have the neutrophils that undergo phagocytosis. So these are the first responders that quickly arrive at the infection site to engulf and destroy the pathogens. The macrophages, which plays a role in phagocytosis and antigen presentation. So after engulfing a pathogen, they break it down and presents its part to the surface to activate their immune cells. The dendritic cells does antigen presentation and phagocytosis. So the most efficient APCs are the dendritic cells. They migrate to the lymph nodes to present antigens to the T cells, acting as the primary messenger between the systems. And the natural killer cells, the lysis of viral infected cells. So they identify and destroy host cells that have been compromised by viruses or become cancerous. So proteins eaten by the antigen presenting cells are broken down to small pieces which are loaded on special receptors like the MHCs and transported to the cell surface. So peptide loaded MHC complex can be recognized by the T cells, and that interaction can lead to adaptive immune response. So adaptive immunity, it's precise and powerful defense mechanism. It's able to better recognize, eliminate and remember the invading pathogen. Development is independent upon earlier innate pathways, and adaptive immunity provides a second and more comprehensive line of defense based on the struggles of the innate immunity. Now the components of the adaptive immune system, like the cellular components, includes the APC, and the molecular components have the major histocompatibility complex, that's the MHCs. So cellular components, that are the APCs, and so what are the APCs? These cells eats the pathogens. It's like acts as macrophages and displays the fragments of them like antigens on the surface to show other immune cells what to attack. The T cells is a type of white blood cells that plays a central role. They can either directly kill the infected cells, that is the cytotoxic C cells, or coordinate the overall response by the helper T cells. And the molecular components, you have the MHCs that can be classified into two ways. Class 1, which is generally found on almost all nucleated cells. It presents the internal cellular samples, and MHC2, which is found on the APCs. It presents external pathogens that have been consumed. You also have the T cell receptor, the TCR. So this is a lock on the surface of a T cell. It is designed to bind specifically to a key held by the MHC molecule. The next one is the B cells, and so what happens is the B cell, this is the membrane bound form of an immunoglobulin, the soluble form of the immunoglobulin released by the activated B cells into the bloods and the tissues. Okay. So this is a more or less an overview of the adaptive immune response that we basically discussed, starting with the dendritic cells, the development of the CD8 and CD4 T cells, that's the You can classify them as the cytotoxic T cell, the TCR, and the helper T cells. So from the cytotoxic T cells, the virus infected abnormal cell with antigenic peptide loaded on MHC1, and the B cell that's antigen loaded on MHC2. So MHC1 it leads to killing of the infected cells, and in the B cell you have the plasma cell, the antibody producing the B cell, the antibody and the neutralization via the induction of phagocytosis. So now we get into the TCR, BCR and the antibody. So TCR is the T cell receptor, BCR is the B cell receptor, and you have heard about the antibody. So structure wise, the T cell receptor is a heterodimer, typically alpha and beta chains, whereas the antibody or a B cell receptor is Y-shaped tetramer, two heavy and two light chains. So the first image that you see is of a T cell receptor, and the next one is a B cell receptor. So the variable regions is located on the N-terminus responsible for antigen specificity for a T cell receptor, and the constant region over there is for the support of the structure and anchoring it to the membrane. So the bonding is the interchain disulfide bonds connected to the alpha and the beta chains that you see over here. And it always is membrane bound, includes the transmembrane region and the cytoplasmic tail. That's about the T cell receptor. For a B cell receptor on an antibody, it's Y-shaped, two heavy chains and two light chains. Located at the tip of the Y is the variable region that forms the antigen binding site. So the constant region forms the stem and the lower arms of the antibody, and the disulfide bonds connects the heavy to heavy and the heavy to light chains. And B cell receptor is membrane bound with a stalk or a tail, and antibodies are the secreted soluble forms. This is basically the structure of the immunoglobulin fold and the recognition of the antigen by the antibody. So you can see over here how the antigen is being captured by the antibody. These are the different antibody structures. You have heard about it. This is not that important. It's the same thing again and again. Okay. We can talk a little bit about the molecular basis of antibody diversity. So what we see over here is antibodies consists of two types of polypeptide chains, the light chains and the heavy chains. The diversity is generated by randomly mixing and matching the different gene segments during B cell development. So light chain recombination, the light chain is simpler and involves two types of variable segments, the variable and the joining segments, that's V and J. So process a specific V segment is randomly joined to a J segment and leads to VJ joining. The intervening DNA is deleted, and this rearranged DNA is then transcribed into RNA, spliced to include the C region, that's the constant region, and translated into a light chain protein. For the heavy chain recombination, you have the variable diversity and the joining segments. First, the D segment or the diversity segment joins the J segment, and then the V segment joins this, so leading to VDJ joining. And this results like a light chain. This rearranged DNA is transcribed and spliced with the constant region to form the functional heavy chain protein. So why does it matter? Because by combining the different VDJ segments, the body can produce over 100 billion different antibody variations, and this allows the immune system to recognize almost any foreign pathogen it encounters. But apart from this VDJ recombination, there are other two types, that's the somatic hypermutation and the class switching. So somatic hypermutation is a later, so first what happens, the primary mechanism that rearranges the gene segments to create a variety of unique antigen receptors is the VDJ recombination. So a later process that introduces the high frequency point mutations into a rearranged antibody genes to define the antigen affinity is through somatic hypermutation. And then a mechanism that changes the constant region of the antibody, altering the effector function while maintaining the antigen specificity, is through class switching. Now, a little bit on drugs versus vaccine. So, drugs are used to treat, manage diseases and conditions that have already occurred. Vaccines are quite preventive. And drugs usually work directly by targeting the bacteria, virus, or the body's processes to treat or manage the diseases. But vaccine strains our immune system to face various existing disease causing agents, generates memory against the specific pathogens. And so the first infection is a slow response. Pathogens multiply, symptoms arise, disease causes, then it leads to memory. And the second infection, there is a fast response. The pathogens are disposed and there is no disease. That's about the drug. But for vaccination, there is no disease. There is a memory that is developed, but there are natural infections that gets fast responses, pathogens disposed, and no diseases. So vaccination protects us from infection by inducing the adaptive immune responses, but bypassing the need for a natural infection. But we need to also understand that vaccination does not mean that the disease will not occur at all, which is a wrong concept altogether. So vaccination is an ongoing global effort. It has yielded some of the most successful stories worldwide. So the last case of naturally acquired case of smallpox was in 1977. Consequences of eradication, universal vaccination not required. And another term is the herd immunity. So if a critical mass of people acquire protective immunity, they can serve as a buffer for the rest. So a little bit on immunotherapy. So immunotherapy is a strategy that uses the body's immune system to fight the diseases, especially cancer. It works by either stimulating the immune system to work harder or by providing some key components like the antibodies, proteins that help the immune system to identify and attack the abnormal cells. You have the monoclonal antibodies, which are mostly produced molecules designed to act as antibodies to target the specific cells. And there are some immune checkpoint inhibitors, which are also a type of immunotherapy that helps the immune system recognize and attack the cancer cells. Right? Okay. So that's all from the lecture part. If you have any questions, you can ask me. Any questions? Maybe I will give 2 to 3 minutes of time. If you have any questions, you can ask me. No questions? Okay. Then I think we can start with the uh, quizzes. So the first one is a match the following. So you people can give answers to this in the chat box. So you have to match versus the types of cells, and over there um, the presence of the particular protein. So let's see, B. Okay, so the cytotoxic T cells are characterized by the presence of CD8 glycoprotein, helper T cells CD4, so A2B1. The plasma cells are fully differentiated B cells, acts as antibody factories, so C4. So A2B1, C4, and the dendritic cells are present. So process the antigens and present them on the MHC2 molecules to activate the helper T cells. So A2, B1, C4, and D3. That's B. Correct. Okay. The next one. Which of the following statements are correct? Macrophages are antigenic antigenic peptides only through MHC1. Dendritic cells express MHC2. Then you have both macrophages and dendritic cells express MHC1 and MHC2. Both dendritic cells and macrophages express only MHC2. Okay, so C, D. So the first one is incorrect. So macrophages are uh, APCs. So while they do use MHC1, their primary specialized role is to present antigens via MHC2. The next one is correct, which says that they express MHC2. The third one is also correct. So MHC1 is expressed by almost all nucleated cells, and MHC2 is expressed specifically by the dendritic cells, macrophages, and the B cells. And the last one is incorrect. Both dendritic cells and macrophages express only MHC2. So it's only 2 and 3. That is option C. Going on to the next one, which of the following immune cell types can perform phagocytosis? So which of the following immune cell types can perform phagocytosis? Quite simple. Okay, I have got one answer. So the cells that can perform phagocytosis are macrophages, dendritic cells, and the neutrophils. The NK cells are non-phagocytic in nature. So it is option B, 1, 2, and 4. Getting on to the next one, which of the following processes are involved in the formation of antibody diversity? So we are talking about antibody diversity. So which of the following processes are involved? We discussed about it just now. B, okay. Right. So the processes are VDJ recombination. You have the class switching and the somatic hypermutation. So it is 1, 2, and 3. That is option B. Question 5. The antibody drug conjugate enfortumab vedotin is specific to which of the following molecules? Nectin 4, nectin 1, cadherin 1, and nectin 3. But before that, okay, so we did not discuss about the nectins and the cadherins. But that's fine. So enfortumab vedotin is a antibody drug conjugate that's comprised of a fully human monoclonal antibody and microtubule disrupting the chemotherapeutic agent used in treatment of advanced and metastatic urothelial cancer. And it belongs to the class of monoclonal antibodies. You apart from this type of antibody drug conjugates, there are others like trastuzumab deruxtecan, you have trastuzumab emtansine, you have brentuximab vedotin. So there are things like that. So basically the pathway of enfortumab vedotin, it is a first-in-class antibody drug conjugate designed to target nectin-4. It's a cell surface protein that's highly expressed in solid tumors, particularly for bladder cancer. It consists of fully human monoclonal antibody attached to a microtubule disrupting agent called the MMAE, that's the monomethyl auristatin E. Now, how it functions, so once the antibody binds to nectin-4, only the cancer cell surface, the interconjugate is internalized. The linker is then cleaved, releasing the MMAE, which inhibits tubulin polymerization and leads to programmed cell death or apoptosis. Fine. So the correct answer is option A. Let's get to question number 6. Okay, so which of the following is true for the process of vaccination? Simple. Which of the following is true for vaccination? So let's see the first one. It says it's intended to form a reserve of memory immune cells for a pathogen. So this is the primary goal of vaccination actually, is to create the memory B and the T cells to remember the pathogen so that when you are exposed in the future, it can respond faster. The next one says it initiates formation of adaptive immune response against a pathogen by bypassing the need of a prior natural infection. This is also true. The third point is incorrect. So while the innate immune system is a first line of defense, body's first line of defense, and reacts to the vaccine injection, the specific purpose and process of vaccination are defined by the development of long-term adaptive immunity. And the last one, these are antibodies used as therapeutic measure against infectious pathogens. So this statement describes a passive immunization, but vaccination is a form of active immunization where the body is promoted to make its own antibodies. Okay, the next one. What order of protein structure does antibodies represent? So antibodies represent what type of a protein structure? So the correct answer is quaternary structure. So most antibodies are complex proteins made of 4 polypeptide chains, 2 identical heavy chains and 2 identical light chains. Because they consist of multiple polypeptide subunits folded together, they are an example of quaternary structure. Right? Okay. The next one, while origin and maturation of B cells takes place in the dash, origin of T cells takes place in the bone marrow and get matured in dash. So what are these two lines? So the origin site for a B cell and a T cell is the bone marrow. So while origin and maturation of B cells takes place in the bone marrow, origin of T cells takes place in the bone marrow and they mature in the thymus. So the right answer is bone marrow and thymus. That is option B. And the last two questions, it says a plasma cell secretes what? What does a plasma cell secrete? So what is a plasma cell? They are fully differentiated B cells. They're fully differentiated B cells. And what do they produce? So these type of cells are specialized to produce and secrete massive amounts of single type of antibody, immunoglobulin, that specifically targets one particular antigen or the epitope. So antibody of a single specificity is what is secreted by the plasma cell. That is option A. And the last one says, which of the following cells link between innate and adaptive immune response? So yes, it's the dendritic cells, which are the professional antigen presenting cells. They capture the pathogens, that's the part of the innate response, process them and then travel to the lymph nodes to present those antigens to T cells, thereby triggering the adaptive immune response. Right? Okay, that's all from my part. If you have any questions, you can ask me. Any questions from previous classes, this class, assignments? If you have any questions, you can ask. Okay then. Thank you for joining. We will meet on Friday, this coming Friday at 6 p.m. And I think there will also be one extra session, a special session on Tuesday, that is on the 7th from 6 p.m. also. So try to join, bring along if you know any friends, colleagues who are taking this course, because that will be a discussion on the entire of what has been talked about. If you have missed something, then you can ask over there, a special session like that. Okay, if you are interested, feel free to join in that class as well. Okay, yeah, fine enough then let's call it a day. Have a good time. Bye.